So could people please take their seats? Good afternoon. For those of you who don't know, my name is Trevor Kilpatrick. I'm Director of the University of Melbourne's Neuroscience Institute, and together with Jeff Donnan, who's Director of the Florey Institute of Neuroscience and Mental Health, we'd like to welcome you to the 2016 Melbourne Brain Symposium. Now, this event marks the 5th anniversary of our occupancy of this building and our sister facilities at the Austin and Royal Melbourne Hospitals. And as such, the event really represents a celebration of 5 years of great achievements in clinically and fundamental-oriented neuroscience research. And I think that, as such, we can really reflect on some really great events and linkages. And those linkages have been with both other sister institutes and also with other academic disciplines. And the program today reflects some of those linkages. I'm really pleased and very grateful to our international and interstate speakers who've been able to attend today, to take time out of their busy set schedules, to Professor Mu-ming Poo, Professor Read Montague, Professor Carolyn Sue, and to Professor Linda Richards. And I'm really looking forward to your talks. You'll see that we've refined the program this year, and the Symposium Committee has worked tirelessly on your behalf to deliver what we think will be a cutting-edge and really action-packed and balanced program, one which is well-suited to be delivered in a crisp afternoon-only format, and there'll be ample opportunity for socialisation at the end of the program with drinks and canopies outside. One of the developments that I'm very pleased to highlight today is the inaugural Mendelsohn Student Lecture, which serves two purposes. Firstly, it serves to honour excellence amongst our graduate researchers, and also it honours a great scientist, Professor Fred Mendelsohn, a previous director of the Howard Florey Institute. I'm also pleased that we've had such engagement with the symposium this year with over 500 registrants. Now, with that, obviously, there are some potential challenges, and it may be during the afternoon at certain stages that the auditorium is overflowing. And this is just to announce that there is also streaming upstairs in the 5th floor seminar room if people can't find a seat. So in essence, I'm really looking forward to a great day. And to move on, I also have the privilege to introduce our first speaker, who actually really needs very little introduction to you. Professor Mu-ming Poo received his Bachelor of Science in Taiwan before undertaking his PhD at Johns Hopkins. He has served on faculty of several esteemed institutions, including UC Irvine, Yale, Columbia, and UCSD. In 2000, he moved to the University of California, Berkeley, where he has served as the head of neurobiology, but most significantly around that time, he also took up an appointment as the founding director of the Institute of Neuroscience, part of the Chinese Academy of Sciences in Shanghai. And since 2014, he's been director of the CAS Centre for Excellence in Brain Science and Intelligence Technology. He's received numerous awards and prizes, but predominant amongst those is the 2016 Gruber Neuroscience Prize, and he's a member of the US National Academy of Sciences. Put simply, Mu-ming Poo is a world-leading authority, and I don't use those words lightly, on axon guidance mechanisms and neurotrophin and activity-dependent synaptic plasticity. And the title of his talk today reflects that expertise, being Neuroplasticity from Synapses to Cognition. Please join me in providing a warm welcome to Professor Mu-ming.
Thank you, Trevor, for the nice introduction. It's really my great pleasure to be here, standing in this audience the second time. I remember a few years ago, another occasion. My talk today will be on the problem of neuroplasticity, I think the most important property of the brain. Unlike the other tissue, the brain is not only trying to achieve homeostasis, it also can change its state into a new state in response to experience. This is the so-called plasticity. And it happens at the synapse level up to cognitive level. So in general, plasticity comes about by activity. When you have an electrical activity associated with sensory processing, motor activity, or cognitive experience, the activity that created in the brain will change the state of the brain, change the structure and function of the neurons and the synapses. And that's what we call this new state, we call this property of plasticity. Now, once you change a new state, then you have learning and memory, the imprint of your experience into the structure and function in the brain, and you have a different behavior response when you, again, meet in response to the environment, you have new changes in cognition behavior. So the question is, over the past decades, is how does activity change the brain, neuronal and synaptic structure? Now, the earliest person who really think deeply into this problem is Donald Hebb. In fact, a Canadian psychologist in the early 20th century, he has been working on various behavioral studies in mice. He wrote a book in the end of his career, 1949. In this book, he postulated that the activity in the brain, the associative activity between neurons, between neurons, if they are correlated in their activity pattern, then the synapses between them are strengthened. If they are not correlated, maybe they are weakened. In fact, this weakening of synapses was postulated by Gunther Stein later. So that leads to the idea that cells are fire together, wire together. This very popular idea of learning rule. Learning always associates the correlated activity in some parts of the brain. So if you correlate that activity, the connection becomes strengthened. This is a very useful hypothesis. For example, let's say we have a memory of a circle. How do we have a perceptual memory of circle? The circular stimulus in your visual field will excites a group of neurons. I wonder whether say no pointer. A group of neurons in your visual system that would make the linkage, the activity, connectivity between these neurons strengthen. So this group are linked together by activity. Now, thank you very much. So this linkage create a set of neurons that are particularly excited by this experience. We call this, Hebb called this assembly. These cell assembly are created by a perceptual experience. So once this connectivity are established, a partial stimulus, we're able to create the activity of the entire network, entire assembly. So you can have a recall of the entire circle by partial stimulus. And this is a simple cell assembly ideas that go along with Hebb's idea that cell connectivity are strengthened by correlated activity. These assembly cells are correlated firing during the experience. Now you can extend these Hebb's ideas to more broadly. For let's say a binding of visual features of an object. Now, this object any object will have many features. There's its motion, its color, its brightness, its topology of the object. All these are part of feature of this object. They are each connected, each set up their own assembly. And this assembly of different features could be linked together because of the presence, constant presence of the object. Then the entire super assembly of all these feature of assemblies of neurons responding to this particular feature together with code for an object, that perceptual memory of an object. And you can extend it to the memory of a grandmother's face. There would be a similar, each grandmother's face have a different features. Each feature was coded by assembly. Together, the presence of grandmother's face eventually set up a super assembly of cells that responding when your grandmother's face is, when you recall your grandmother's face. Now, one can even extend further, I mean, this is all hypothesis, extend further that concept of grandmother, including many aspects, including grandmother's face, grandmother's song, her the stories she told. Everything associated with grandmother, including her names, are part of the grandmother concept. Now, each of these features or component of grandmother's concept are coded into assembly, super assembly. And these assembly are put together in the brain. And that brain, that component is probably in different regions of the brain, right? Names probably in the language region, face areas, coding for the grandmother's face. All these are super assembly everywhere in the brain. And these assemblies are linked together in the presence of grandmother early in your life. Eventually you form the concept of grandmother. This is a hypothesis. One would argue that strengthening of long-range connection between different parts of the brain might be responsible for setting up this concept of grandmother. Now one can say, well, the recall of the grandmother's face, you will see her name, you recall the entire grandmother's feature, re-trigger the entire assembly of the concept. This is I think this is probably the most to me the best hypothesis about how our brain codes for memories, including more higher abstract ideas of concept. Now, obviously, this is a hypothesis that needs to be tested. One can start to work on this, and some of the work is going on in my own lab on this. Now, one can also look at the development of the circuits using Hebb's idea. In early development, the molecular cues in the retina and the tectums set up the early connection which are diffuse and not quite precise. Then experience will sharpen the connection to one-to-one connection, retina corresponding to tectum, more precisely mapped connectivity. Now, this activity also can this activity-induced refinement can be explained by Hebb's idea because the connections made by nearby neurons, nearby retinal neurons, are likely to be excited by an object, some visual stimulus, usually spatially and temporally correlated. They tend to excite nearby ganglion cells, so their activity pattern, their connectivity to tectum cell are stabilized. While long-range, other uncorrelated retinal neuron, their connectivity will be eliminated by the idea of cells fire together, wire together. Right? So this is a simple idea that can explain even the development of the map, starting from a crude map into a fine-grained map in the visual system. So a few years ago, many years ago, 15, 20 years ago actually, we thought the Hebb's idea could be tested more precisely. We talk about correlated firing of nearby neurons, but how correlated, we don't know. So we can actually record it from the tadpole of a xenopus, from a tactile cells that receive input from two regions of the retina. And we can excite retinal neurons, get a response from the tactile cell. And we ask how correlated have to be for the retinal input to stabilize their connection into a single tactile cell. Right? If they are uncorrelated, would they compete with each other and lose the connection? So quantitatively, how does the Hebb system work? What we found out is we're actually quite surprising. Now, when there are two inputs, one retinal input gives a subthreshold depolarization, the second input gives a superthreshold excitation, fire spike in the tactile cell. If the first input comes before the spikes, you get potential, you actually strengthen. If the input comes after the other input has fired the cell, fired the tactile cell, it will actually become depressed. Now these two input 1, 2, they are all spaced 10 milliseconds apart. But when input comes before the spike, you potentiate. Input comes after the spike, become depressed. They're all similarly correlated, but timing of spiking in the pre- and postsynaptic cell is critical. Right? So that argues that, that the Hebb's idea of simple correlation are not enough. You have to talk about which one fires first when two cells are linked together. Pre before post is LTP, post before pre is LTD. And it all happen within this critical window of tens of milliseconds. It's like competition of input to a cell. Now people, the input comes in and compete with each other. Those who are become potentiated are those input that actually comes before cell fires. It contribute to the firing by contributing depolarization. Those input wins, gets rewarded. If it comes a bit late, when other input have already fired the cell, it's too late to help. The postsynaptic cell excitability gets depressed. So it's like a competition among nearly equal. They all comes within tens of milliseconds. Which one wins are those gets there earlier. Now, the input that comes really early or really late are not in the game. They're not competing. Like any other game, it's all competition between nearly equal. So this idea leads to a revised Hebb's learning rule that says that the correlated activity can be either strengthening or weakening the synapse. It's the pre before post firing is strengthening, post before pre is weakening. And it's called spike timing dependent plasticity. I mean, this finding actually has also been observed a year earlier by Henry Marquand and Bert Sackman in their study of visual in the cortex. Well, the back-propagating action potentials from the dendrites, if it comes before presynaptic spiking, it suppresses the synapse. If it comes after, it strengthens. So these are very similar. But here we have an in vivo condition where the input comes in by stimulating the retina, a physiological competition. Condition for competition also show this spike time independent plasticity. So let's see, let's do a thought experiment now. We have a set of neurons which are connected to, in beginning, before we have learned experience. They are connected to each other. The connection could be both ways. So when, they could be recurrent connection. Each neuron are both sending to and receive connection from any other neuron. And if you think about this, each unit become a set because each unit here is a set of group of neurons. Then one group of neurons can excite next group, so we can have a chain of excitation. So let's think about experiment. The Duncan experiment, where we now align this our stimulus so that we are firing in a sequence, firing always in this sequence, this cell first and then next cell. So a sequential excitation rather than a simultaneous excitation of the entire net, like the earlier example I showed. This is a sequential firing of a set of neurons, a cell assembly. And the result of this is all the downstream connection are all excited because the cell fire before, for every downstream synapse, the cell fires pre first and then post. Pre-post, so you get strengthening of all the downstream connections. All the upstream connection is reversed in the firing, so they are all suppressed, become weakened. So after a preferential sequential excitation of a set of neurons, what you get is that you have stored information in the strengthening of synapse only in one direction. You have a sequential excitation, strengthening of the synapse. So once you have that, now you have an asymmetric connection among the set of neurons. You can now use this stored information as a storage of sequence information. If you then fire only the beginning, you just fire the beginning neuron, so excite this neuron, because this group of neurons can excite next one and next one, so you can actually trigger sequential firing. A partial stimulus in the beginning of this set of neurons can trigger a recall of a sequence firing. It's just like you remember a tune. A tune, you can remember the entire tune by giving you a few notes in the beginning, because that tune is a sequential memory in your brain. Now, if you give you the last few notes, you cannot recall. the sequence. The sequence for recall the sequence of a tune has to be in the same sequence as your storage, right? Because that can awake this entire net cell assembly in one direction. So this is a thought experiment. Can we demonstrate this in the brain? So when we have carried out some experiment recently, just by putting multi-electrodes in the visual cortex, so that we ask, can we store sequence information in the visual cortex by giving in the visual field a moving ball across the visual field, which excite a group of the neurons in that recorded by this set of electrode. So this is exciting a ball moving across the receptive fields of this visual neuron recorded by the electrode. And so we have a conditioning, only repetitive excitation, a sequential excitation. Then we ask, can the visual cortex store this sequence information? Can we recall this sequence firing by only giving the initial stimulus, right? Like called a thought experiment. And this is an experiment, and we represent the firing of recorded by each electrode. This is receptive field by the 16 electrode set, and then we sweep across the visual field repetitively to excite this group of neurons, and the firing is coded by the brightness. Right? So you see a sequential firing when you sweep the ball across the field. So after training, conditioning for a few times, and then you ask the question, can you now recall this sequence by firing, by only exciting, only give a flash at the beginning position, or a flash at the end position, and which one triggers more sequential firing recorded by a neuron? The answer is there are more sequential firing triggered if you flash in the beginning point than you flash at end points. That means that you can trigger the firing. The information of sequential firing is stored in the neuron. This is one of the examples. So 16 electrodes record a roster of spiking. They are more or less random. There are a lot of spontaneous activity going on. Flashing at a starting point before the conditioning shows no sequential firing of the recorded by the electrode. But now you give a sequential stimulus of a ball, and the 16 electrode fire in sequential manner every time you sweep the ball across the visual field. So after this conditioning, now let's flash again in the beginning and see if we can see sequential firing. And this is a flash again in the starting point. Now out of 7 trials here, you see at least 4 or 5 trials with sequential firing appearing. Clearly different from the before conditioning. So this is the short-term memory of sequence spiking within the visual cortex. We think that this is between the intracortical connections that's been strengthened in unidirectional manner because of the sweeping across. We can also show that by optogenetic stimulation. Optogenetic sweeping excite a group of neurons in the visual cortex, V1 neurons. They can also store information, sequence information, just like this example. So this is learning. Learning of sequence memory can be accomplished by Hebb's idea of strengthening of synapse, but in a temporally specific way. So you have spike time independent plasticity used by the cortical neurons to store sequence information. All the memory, episodic memory, usually have sequence. Well, we remember what we do today. We did this first and then the other. It's all have a sequence. Right? We remember the music, the sound, all this sequential information. So being able to code sequential information by a synaptic level learning rule of spike time independent plasticity is very useful, very rewarding. So, but how much do we know about learning and memory? Where are the memories stored? We said we can create some memory in the visual cortex, but they are very short term. After a while, it disappears. Well, a few minutes with sweeping balls we can have a sequential firing recall from the cortex only for a few minutes and then disappear. Right? Where are the learning of memory? It turns out that we know not much about the learning. Where is the memory storage? This is a question asked for almost a century now. Where is the engram? Where do we store the memory in the brain? We actually, which brain region store a particular memory? We don't know very much. And which neuron, which synapse store memory? We don't know much. Right? So with still open question, how is the memory stored? Now we know something pretty good at the synapse. We know the LTP and LTD, long-term potentiation and depression created by the activity. We know that must be a mechanism. If you prevent LTP and LTD to happen, the rat would lose, mouse would lose its long-term memory in the brain. Right? They really require synaptic change that's long-term, which is LTP and LTD. This is fine, but is there any structural change? Is the connectivity between neurons actually modified? If new forms, new synapses form or old synapse eliminated by learning? We don't know quite well whether that's true yet. We don't know how the memory is retrieved, and the idea is that maybe if we reactivate the synapse that store the memory, we're actually retrieving. As long as those synapses are active, that's where the memory reappears in our brain. I mean, that's the most popular way of thinking about this. How does memory decay with age when we have dementia or Alzheimer's? Does it, the ability to form memories reduced, or is it the stability of the memory that we create are low, changed? All these are very outstanding questions in the brain. Basically, we know very little about learning and memory, and that's really a lot of open questions to study. Now, I want to address the question about whether the structure has changed. Now, most of the excitatory synapse is made by axon terminal at Bouton, ending on the synaptic spines of on the dendrites. And this particular structure is very, very useful because the changing of spine structure, the neck thickness or this structure can control the current flow generated by the terminal. So the amount of weight, this influence on the dendritic depolarization can be changed by changing the spine structure. So spine might be a useful way of controlling the synaptic weight. Right? So the question is also, if the memory is stored in this type of structure, do we form new spines and new boutons when we form new memory? And that's always a quite interesting question. If you look at LTP, when you artificially create LTPs, you can find more spines are formed on the dendrites, suggesting there are more synapses formed. But is that really true in our brain when we form a new memory? And that needs to be demonstrated. So we recently did some experiments because this is possible to do now in living mice using 2-photon images of the brain structure, where you can look at the individual dendrites in the brain with an open window, and that you can monitor it for a long period of time, days and weeks, on the same dendrite, same spine, and you ask what happened to those connectivity when you learn. Now the question is, you have to find a place to look for this. You don't know where they are, the memory. Right? So we look for a very robust auditory field conditioning. That is, you give a tone to a CS, a conditioned stimulus, a tone of a particular frequency, and then at the end of the tone, you shock the mice with the electrical pulse. So tone shock are paired for a few times. Then when you just give it a tone, the mouse will freeze. Afraid of a shock coming. So the field memory is there. So this is a very robust way of creating memory. The control is that if you do the same shock and tone, but unpaired, random, and you don't get a freezing response. Only when they are paired, you get a freezing response. Highly reproduced by over 90% of the cases, if you just give a sound, the mouse would freeze. So very robust field conditioning. This has become a very useful model system for studying memory, especially long-term memory in the brain. And the circuits for this is well worked out. Over the years, people have worked out the importance of amygdala. That receive sensory information from the CS, the auditory signal, or sound signal, or visual signal. It can all be the CS. The US unconditioned stimulus is a shock. This shock is also go through the somatosensory pen processing system. They converge in amygdala. Then the amygdala sends out output to control the freezing response when they only hear the auditory signal. So now what I want to show you today is that we found that from this is a classic circuit. We found from the LA, the lateral amygdala, there's an output that goes directly to the auditory cortex. And this synapse at the auditory cortex is necessary for long-term memory, field memory. And we look at auditory cortex because we just want to look at cortex. It's easier to image the cortex by two-photon imaging. We cannot see it too deep with the photon imaging yet. So we are looking for cortex. It turns out that we found out the auditory cortex has changes that are associated with long-term memory. And then we start to look, what are these synapses? It turns out these synapses are coming from amygdala. This is how we discover this new pathway that has not been, has more or less been ignored in the field. So how do we show that auditory cortex is important? We can do a bilateral inhibition of the cortical activity with muscimol, increased inhibition, or APV that block the plasticity, block the LTP, generation of LTP in the auditory cortex. Now, if you do that, the field memory cannot be formed. If you apply this inhibition before conditioning, certainly you cannot form the memory. That may be the generational memory require this auditory cortex. So that's not surprising. But the surprising thing is that if you apply this drug infusion after field conditioning, you can prevent the recall of the memory. That is the conditioning itself creates some memory. This memory you can get very, very good recall. But if you inhibit the auditory cortex activity after conditioning, you cannot retrieve memory. So the storage of the learned information in the auditory cortex is important, and plasticity because it's APV, AMD receptor dependent. Now this connection from from the lateral amygdala to cortex can be demonstrated. You inject, transfect the lateral amygdala neurons, the terminal of amygdala. Axon projections are found in the auditory cortex. A lot of them in the layer 1, where they make synapses with the dendrites of cortical neurons. You can do reverse labeling also. You inject beads, retro beads in the auditory cortex. You can label the lateral amygdala. You can also do the so-called rabies virus tracing to look at presynaptic neurons that make synapse with auditory cortex, layer 5 neurons. You also show that the lateral amygdala neurons are labeled. So that's suggesting there is a direct input from lateral amygdala to the auditory cortex. Now, if you inhibit activity directly, the terminal of this lateral amygdala neuron, so you transfect the channelrhodopsin or the heterodopsin into the lateral amygdala neuron that projects to the auditory cortex, and then you use optogenetic light to excite those heterodopsin so that the axon terminal are inhibited in auditory cortex. Then the freezing response is reduced, not completely wiped out, significantly reduced. Right, so that suggests this is all after the conditioning already established memory. The recall of memory can be reduced by inhibiting this amygdala to auditory axon. You can do another type of inhibition which works actually better. It's called chemogenetic approach. That is, we express a type of a modified G protein into the amygdala neurons, And then you apply a ligand called CNO, a ligand that activates the G protein, that inhibit the activity of these neurons. And in that situation, we have a more prolonged inhibition. If you apply that trick to inhibit this activity of these lateral amygdala neurons, the fear condition is totally reduced. Right? The control, transfecting other thing has no effects. Right? So this is suggesting this pathway is actually required. Activity of this amygdala is required for this long-term memory. So now you can now look at the structure. Now we know that this pathway is important. We can specifically label this axon from amygdala and look at whether they change. Right? So we can watch over a different time, looking at the same region. You can see that for this little bouton made by the amygdala axon in the cortex, you image after you transfect the cell, compare the images before and after conditioning, immediately after conditioning or a few days after conditioning, and you look at what happened to this bouton, the presynaptic boutons, when you store the memory. It turns out that this bouton actually changes with time. Some boutons disappear, like this one. Some new boutons appear. This swelling of the presynaptic axonal bouton usually signifies the formation of synapse with a postsynaptic dendrite. Although we don't have a dendritic labeling here, so you don't see the presynaptic spine, but all these boutons actually has a corresponding spine in contact. So one can look at a few days after condition and see what is the turnover mechanism. Is any new bouton formed when the memory is formed? The answer is yes, there indeed. There's a before conditioning or in the control, there's a small percentage of bouton that disappear or a small new bouton form at a low rate. So this connection in the adult brain actually are dynamic. Not all of them are staying for a long time. Some of them are getting turnover. Right? The turnover rate is a few percent over days, over 1 day to 3 days. The turnover is about 10% at most in 3 days. Now, if you have a conditioning, there are not more bouton formed. Significantly more bouton is formed. Each of these points is actually one set of recordings from one brain. So there's a lot of experiments, right, to count many dendrites, hundreds of dendrites, looking at the percentage of new bouton form. And this is highly reliable, even though you see there's off scattering, but a statistical highly reliable increase in the number of bouton formed by the amygdala terminal. But the elimination of this disappearance of the bouton didn't change very much. So you have the eliminations remain constant, formations increased after memory, so you have a net number increase of this connectivity. Now, if you look at another input from a prefrontal cortex, labeled prefrontal neuron, their terminal in this auditory cortex doesn't change. So there's a specific increase of the amygdala terminal. Now you can also label the postsynaptic neuron with fluorescence. So you can look at layer 5 neuron or layer 3 neuron, look at a dendrite, and look at whether dendritic spine also changed. The finding is very clear that there's also a corresponding increase within 3 days after field memory, an increase in the spine number. New spine are formed. The elimination of spine also occurs at a constant rate. That doesn't change. So you have a corresponding to the increase in the presynaptic bouton, the postsynaptic spine of the cortical neuron, pyramidal neurons also increased. Right? If you look at the layer 2-3 cells, there's no change. So it's a layer 5 neuron, layer dendrite. The spine on those dendrites are selectively increased. So now this is all pre and post labeled separately. The key is to really look at the Both sides are labeled and look at the contact, whether that synapse is changed. Now, this is harder to do. In the field, people are trying very difficult for a long time. We finally succeeded in doing a pre and post synaptic label. Pre-label with green, with this amygdala axon, post-label with red. So here you can see a synapse, bouton and spine. This is stable over a 3-day period. But there are new spine formed, new synapse formed. For example, here, there's new bouton appear to form contact with the spines. Right? So that's happened in 3 days. Some synapses are eliminated. In this case, the spine disappeared after 3 days. So you can look at pairs of pre- and post-synaptic elements and look at a specific amygdala to cortical neuronal synapse. Are they changing with time? So after looking at these pairs, we finally had this summary that the amygdala to cortical synaptic pairs increases significantly. Even actually after 2 hours, you can see significant increase. Now, the 2 hours in our separate studies of spine and boutons, they didn't change very much, they're not significant. But with looking at pairs, they become significant. So there's clearly increase in spine formation, but the illumination doesn't change. I mean, look at other synaptic pairs from the thalamus or from the prefrontal cortex. There's no significant change. So it's a specific increase in synapse formation from amygdala. So, and this increase in the amygdala synapse, the pairs, the increases correlate well with the freezing level, correlate with memory. Now, finally, the most interesting finding to me is the following, that this new synapse that's formed are not totally new. They are synapse formed on the old synaptic pair. Now here is an example. We have the first type of form. There is a synapse already there. This is a bouton. The postsynaptic spine are not labeled in this case. But in this particular neuron, new spine form at this old bouton. Right? So the new elements added in new spine form on old bouton. So a synapse is there. You want to increase connectivity, you add a new element to it. This is adding synaptic, postsynaptic spine. There are another type 2 is adding presynaptic new elements. Here's already a new spine. Let's hear. The spine is there with already forming a synapse with a bouton, but a new boutons appearing to make synapse on the old spine. So adding a new bouton on an old spine or new spine on old bouton, these two types accounts for majority of the synapse, essentially all the synapse. Right? You can look at an EM structure showing that this multi-synapse bouton, more than one spines on the bouton, or multi-synapse spine, a single spine with multiple boutons. This frequency increases with this memory. So de novo formation of new spine doesn't happen in adult brain. Doesn't happen with a very low percentage. Now, out of 500 pairs we look, only a few. It's totally new. But all the rest are the old elements. You add a new component to make the synapse connectivity more efficient. Now, this is reasonable because in the adult brain, it's all very crowded. Everything's jammed together. You want to make a new connection, you better just do it in the most efficient way, just adding a new element to it, so that you make a connection better. Right? So this appears to be how the memory associated with memory associated changes happen in adult brain. So finally, a few minutes left. I want to talk about the cognition. Now, we are looking at very synaptic level. The hope is that this synaptic change reflects cognitive function about memory, the the behavior of the animal. The cognition, we can think about cognition of the outside world, how we perceive the world, the sensory perceptions, the concept of categorized objects in our environment, attention paid to particular stimulus, decision making, all these are the cognitive function about outside world. And this can be studied in many animals, from flies to mouse to primates, all of them. There are many animal models for this. But for a higher level cognition about self and non-self, the self-awareness, empathy, theory of mind, knowing what other people are thinking, this probably only occurs in humans and a few species of primate. Now, there's debatable whether in which primate you have empathy, you have theory of mind. Now, people think they have demonstrated theory of mind for chimps, but it turns out not true. Anyway, so this has happened. If you want to study this higher cognitive function, which is more relevant to humans, we have to study at a primate, at least a primate level. Obviously, language is only very unique. Once you have an awareness of self and non-self, you can develop communications, and those communications eventually become language, which is really the highest function of any cognition, and it's probably unique to humans, right? So all this depends to a large extent on learning and memory of the plasticity of the brain. So how do we study this higher cognitive function? Some of the higher cognitive function is very relevant to brain disease, like autism, schizophrenia. All these are having to do with global functions of the brain. So, for example, autism. So can we develop animal model for autism? Now, there are mice models, but it's a stretch to talk about mice with autistic behavior, like social interactions. So we attempted to find models for the monkey, and two labs in our institute, Zilong Chiu's lab and Sun's lab, they collaborate together, develop a transgenic overexpression model using human MECP2, a protein relevant to Rett syndrome. Overexpressed MECP2 in monkey produces the first generation of monkeys which has an autistic phenotype. And the second generation, which we can also produce, which comes from the first generation, the male of the first generation, is actually, there's a germline transmission. A lot of the transgenes are actually inherited in the second generation monkey in different chromosomes. Right, so we have a transgene propagation in this model, which is very nice. This transgenic monkey has autistic phenotype showing about the repetitive behavior. They do this circulating motions in the cage a lot more often than the wild-type animals. But they like to go around in the same direction, actually, in a cage. Right? This is a repetitive motion. They also have increased anxiety. If you look at them, they tend to have calls and sound produced that reflect anxiety, higher anxiety. And the most important is this social interaction. You'll have two monkeys sitting in the cage. You'll find that the transgenic monkeys interact much less with the wild-type monkey or with all other monkeys, including transgenic. Significantly reduced interaction by sitting together and grooming each other, all this type of normal social behavior. So we have this autism model. What can we do? Once we have the autism model, we can go for mechanistic study. What changes happen in the brain when we have this autistic behavior? We can brain imaging, all the other. In the long run, I might even think about, can we correct this autistic phenotype by some sort of a training? Intervention, right? So can we actually do intervention in a primate that reveal the result of intervention that's higher cognitive function that's more unique to human? So here is another example, this self-awareness. It's thought to be a human trait, only happen for a few species of ape that can look at a mirror and then show behavior that seems to be aware of themselves, scratching their face, looking at their teeth in a mirror. And a human baby does this 2 years on average after 2 years of age. If you have a kid, you can watch him. Some study 1.5 years, some average 2 years. Takes 2 years for the baby to know what they see in the mirror are themselves. Now, why? Is in the program development of the brain so that the baby become aware of themselves, or is it taught? Is it learned? Is the parents taught them that the image in the mirror are themselves? Right? So these are our questions. So the question, and also autistic patients seems to lost their self-awareness. Some of them show no awareness when you give them a mirror. Maybe if we have a model where we can investigate this mirror self-recognition behavior. So last couple of years, a colleague of mine, two students were able to train monkey to show self-awareness. Now, people have, in the field for 50 years, people thought monkey show no mirror self-recognition. There are a lot of studies in primatologists. They were able to train monkey to do this. But we developed a trick to train monkey. That is to have monkey sit in front of the mirror. Here's the monkey sitting here looking at the mirror. And we train to associate the image on their face with some sensation. This is given a laser pointer with a little bit higher power so that they feel tinkling feelings on their face. And then associate the image in the mirror and the feeling in the face. Now, by doing this for a few weeks, you have to feed them just to encourage. Now, in the beginning, you have to encourage them to do this, but after a few weeks, they know the association. Even for a monkey to sit without use a very low power laser, no feeling, no somatosensory feeling, they would do the touching behavior, even without food. So they seem to be able to associate this image with their face. So once this is done, then we put them in a home cage to do this. We actually have to do this standard mark test. This is all in the field. People put a mark on the face and see if the monkey touch themselves on the mark. So here's a mark on the face. They would touch it, a dye mark on his face. When they look at this, they would touch it, scratch it. This is a standard mark test people use. Once you pass this test, people would judge, primatologists would judge that this monkey have learned mirror self-recognition. Now, so you put the monkey back to the cage. This is totally spontaneous. You will put a mark on the face. They can do this mark test in a cage. No more food, no more encouragement. They will appear to be able to recognize themselves and do the standard behavior of touching, looking, and smelling. This is in the cage. Now, finally, most convincing comes from not even any mark. Look at the spontaneous behavior of the monkey in the cage. So here, this is a trained monkey, this is an untrained monkey. This monkey is pulling no marks, just pulling hairs, seems to be enjoying himself. And looking at places where was never able to see before. The interesting thing, this monkey, now not every monkey can do spontaneous behavior. Now, 5 out of 7 monkeys were able to go to this stage. 2 monkeys, there are stupid monkeys as well. They should never learn to have this. This monkey has been sitting together, or caged together for 2 years, never learned without training. Right? So So what's the implication? The implication is that rhesus monkey can learn mirror self-recognition, some sort of body awareness, right? And then human babies can acquire the ability. Maybe they acquire the ability. We don't know that yet. I think this is something to investigate for the pediatrician to really investigate it. Impairment of self-awareness might be able to be intervened by training to have this association. Okay, my time is up, and I'll stop here. Thank you very much.
You've given us a new definition to monkeying around, really. Look, we are running short of time. I'm sure Mu-ming will be very happy to answer any questions during afternoon tea. I might just take Chairman's privilege just to ask a couple of things quickly. Right. Firstly, the no new spine rule in adults. Do you know if that extends to the circumstance of regeneration post-injury? Right.
Okay. I think we're using adult brain that are not under the trauma of regeneration. There's a reactive sprouting after injury. New spines are formed. A lot more growth is happening during regeneration after injury. Right? So the rule will be different. We're talking about an intact, un-injury condition that this rule probably apply.
And you've convincingly showed us what is necessary in the process of plasticity. I'm wondering whether ultimately it's sufficient. And I'm thinking about the work of Doug Fields in relation to his perspectives about myelination, saltatory conduction, coordination of action potentials, whether you think that's important or whether you think it's just an epiphenomenon.
Right. So it's very difficult. You have a very good question. It's very difficult to demonstrate sufficiency for memory because we don't know where the memory are stored. The best experiments, you know where the memory are stored in what synapse. You block those synapses, memory disappears. You activate those synapses, memory appears. Right? That's the best experiment. Since we don't know where they are, how many areas are involved, we cannot do sufficiency experiment. We are trying to manipulate these pathways if they enhance the memory. But no, that's only part of it. So nobody were able to do sufficiency experiment memory test. All the experiment you hear about blocking LTP affect behavior, blocking this, blocking that, affecting memory. They also necessary component for memory, but nobody were able to do sufficiency experiment because we don't know all the set of places where the memory is stored in order to do the sufficiency test.
Thank you very much for a fantastic talk.
So if I could just echo what Trevor said earlier, to remind you, my name's Jeff Donnan. I'm Director of the Florey Institute of Neuroscience and Mental Health, and we each year hold this wonderful symposium in partnership and celebrate neuroscience, and you can see we're off to a wonderful, wonderful start. What a fabulous talk. And we're in store for another one because I'm going to introduce to you what I call a threatened species, and that is a person, a little like me, who's both a clinician and a busy clinician and a researcher. Carolyn Sue, I've been following her career for many years, not all that many, but many years, from when she trained first at Westmead in mitochondrial disorders, muscle, and then moved to the Royal North Shore Hospital, Sydney University, where she's been following a stellar career in mitochondrial disorders and muscle, but also more broadly the genetics of neurological disease. She's currently the Director of Neurogenetics at Royal North Shore Hospital, and also she is the Director of the Sydney node of the Adult Stem Cell Centre of Australia. So both very important positions. She also co-founded the Australian Mitochondrial Disease Foundation. So she does many things in the limited time she has to do it, as I say, both a clinician and a researcher. So it's an enormous pleasure to welcome you, Carolyn, and to ask you to give your talk, the new one-stop shop, which we're very interested to hear, simplifying the diagnostic pathway using next-generation sequencing in neurological disease. Over to you. A warm welcome.
Well, thank you very much, Jeff, and also to the organisers for inviting me to this wonderful symposium. I'm really enjoying the first talk. I'm really pleased to come back to Melbourne, where I did half of my PhD on mitochondrial genetics. So it's really a very big honour for me to come and speak here today. Well, I hail from Sydney, as Jeff just said. This is our institute here, and I guess what I really want to emphasise is this synergy between clinical research and scientific endeavour, I guess. Here at the Conning Institute, which is where I work, let me see if I can get my pointer here. Not sure I can make it work. It's not going. So this is our Conning Institute. It's co-located to the hospital here, both public and private, and also we're close to the Sydney Harbour. So if you want to come up and see the harbour, then you're very welcome to come and visit. Okay, well, what I want to do is emphasise that Australia is really obsessed with reality TV. And if you're like me, this is one of my favourites, MasterChef, and this is the dreaded cock and bush. And what reality TV, this TV show really, is to convince us that home cooks can actually achieve making this kind of masterpiece here. I don't know if anyone's tried to do this, I haven't, but we're led to believe that if you have the instructions to do this, and here is the recipe for the crock and bush, you can download it from the MasterChef site. I don't have any royalties, unfortunately, but you can actually go ahead and try and make this construction. But, you know, as a neurologist and a clinical researcher, I'm not really interested in making cock and bushes. I'm actually interested in working out how human disease works. And I guess, wouldn't it be nice to know how people like this really worked? If you're not really into science, then perhaps you'd prefer to have, you know, be able to work with, like this guy, the Don. Or perhaps you like to know how these people work and tick. Well, in human life, the blueprints for how we work as humans is really hidden in the human genetic material, the human chromosomes. And here are our 23 pairs of chromosomes lined up here, where we have all of the genomic instructions that make humans work. I'd like to put in a little plug for mitochondria, which is often the forgotten genome. And so mitochondrial DNA is also hidden in the human genome here. It's sort of labelled here with gold staining within the cytoplasm of mitochondria. Well, it's a large endeavour because human genes are made up of DNA, and there are 10 trillion strands of DNA within each human. So if you were to line this up, end on end from your chest to the sun, it goes there and back 4 times. It's a significant amount of information to understand. I'm going to hail back to my PhD here in Melbourne. So this slide was created from my PhD work, which I performed in Melbourne with Ed Byrne, under the direction of Ed Byrne. And you can see here, this is 20 years ago, we could sequence DNA in this sort of black and white structure, later in this radioactive labelled genetic material. And what I was interested in was this little tiny mutation, a small change from A to G. You can see that it's heteroplasmic. This is from mitochondrial DNA at position 3243. And really this caused human disease. This is it in the sequence here. This is the one little change I was looking for. It took me about a week to generate that slide after a couple of months of learning how to do the technique, and it was really determining this, my favourite genetic mutation at 3243. It causes MELAS syndrome, mitochondrial myopathy, encephalopathy, lactic acidosis and stroke-like episodes. Well, enough about that in a moment. Well, more about that in a moment. When I went to my postdoc, we were able to do it all in colour, so you could get 10 times as much sequence in several hours, and so it was a much better improvement. But now we have this new technique, massively parallel sequencing, which is able to sequence massive volumes of genetic material now. We just basically get the DNA, we fragment it all up, you label it. If you want to do just the exomes, you take those and capture those and then sequence. If you want to do whole genome sequence, which is what I like to do, you just take the whole lot and then sequence it and then use huge computer programs to help you analyse it. Well, what are some of the disorders that I'm going to try to talk about? Well, as Jeff told you, I have an interest in mitochondrial disease, and I'd like to take this as an example, although you can use any human disease that you like with this technique. These are a couple of mitochondrial diseases, and the reason why I'm showing you these is because really they're a complex disease. They have many different clinical phenotypes. Here is MELAS syndrome. Again, I can't get my cursor. Okay, so this is MELAS syndrome causing epilepsy and stroke like epilepsy, nerve syndrome, CPO, finger there, Ben, muscle weakness, just trying to touch Mel's finger. Palms up. Get him off out of here. Down. Just really good. Just look up for me. Take your nose. Finally put your head down. Okay, another side. These are just to give you a flavour of the variety of Stay, come back. Close your eyes. Okay. So in 1988, the Queen Square Group published that these mitochondrial diseases were associated with mutations in the mitochondrial genome. Mitochondrial genome, just to remind you, you've got the mitochondria swimming around here in the cytoplasm, and it has its own genetic material, mitochondrial DNA, and that's in addition to the nuclear DNA, where the chromosomal material resides. Of course, mitochondria are intimately related to and under the control of both genomes, nuclear DNA and mitochondrial DNA. And it's because while we have mitochondrial genes contributing to the mitochondrial respiratory chain, we also have a dependency on nuclear genes that are required to assemble and comprise some of the subunits within this respiratory chain. They're also needed to assemble everything, and also there are proteins required to regulate the replication and proofreading of mitochondrial DNA. And we now know that there are over 150 nuclear genes and 150 mitochondrial genes that are mutations that are associated with mitochondrial disease, and this list keeps on growing with next generation sequencing techniques. Now, as I said, mitochondrial disease is clinically challenging to diagnose, and that's because there's phenotypic diversity. I'm going to take you through some of this, but essentially because mitochondria are present in almost every tissue in the body, every part of the tissues except for the red blood cells. So if you have problems in the muscle, brain, or colon, for example, you have different clinical phenotypes depending on the level of heteroplasmic load that you may have from these disorders. Let me just show you some of these issues. This is to explain or demonstrate genotypic diversity. So here I've just taken 5 pictures of 5 patients with chronic progressive external ophthalmoplegia. These are the genetic mutations that they have. You can have mitochondrial DNA deletions, point mutations, and you can have nuclear genes. Here you've got thymidine phosphorylase, OPA1, and Twinkle, all causing the same phenotype, although the genotypes are different. Now, why is this important? Well, it's because different genotypes are associated with different forms of and complications of chronic progressive external ophthalmoplegia. Here you can get cardiac complications with these 2 types, blindness in these 3, respiratory failure in this person, and the treatment is different. So you can use iduribone in Leber's hereditary optic neuropathy, and enzyme replacement is required for Menge syndrome. Also, the inheritance patterns are completely different. You can have sporadic disease, maternally transmitted disease, autosomal recessive or autosomal dominant conditions. So this, so identification of genetic, the causative gene really helps you to personalise the patient's treatment and also inform family planning. Okay, let's talk about the flip side. So these are just 4 patients with the MELAS mutation. Here you can see a patient with a stroke-like episode and external neuroplegia with 3243. This person has stroke, status epilepticus.
Good, and follow it down.
This patient can turn off their pager and a muscle disease.
And follow it across again.
Spinal cord, if I can get my cursor. Parkinsonism associated
Off the bed again.
with the same genetic
Lift up
mutation.
again. Lift off the bed.
Squat down.
Bring your head off the bed. That's it.
No,
These last two were related.
Just do that tight as you can.
Ready?
Good.
Very hard.
Okay, now why do we need to know about this? Well, mitochondrial disease is much more prevalent than we initially thought. When I first started my clinic, now some 25 years ago, then I felt that this was I was taught that this was a really rare disorder that only affected children. But I kept on getting referrals and was supported by many neurologists here present in the room and also others. And so we embarked on prevalence studies to see how common these disorders were in the community. We collaborated with an epidemiology group who did the Blue Mountain study. This was very close to the Westmead campus where I was working at the time. And we were able to extract not only blood but also hair follicles, a tissue of choice for mitochondrial DNA analysis, and was the subject of my thesis down here in Melbourne, to see how prevalent these conditions were. To our surprise, we found that 1 in 500 people in the Blue Mountain studies, community members all over the age of 50, undiagnosed of having any of these related disorders. 1 in 500 of those had the 3243 mutation. We looked at a second mutation, the 1555 mutation. This was associated with what was at that stage thought to be aminoglycoside-induced hearing loss. But we again found this in 1 in 500 community members of the Blue Mountains. So again, a combined prevalence of 1 in 250 people, or about 100,000 Australians. This work has been replicated by other groups, and the UK group also studied a number of different mutations and found again that the population prevalence in the community was laid somewhere between 1 in 200 and 1 in 250 people who were who were mostly healthy. So in this sense, I like to think about mitochondrial disease as being easy to recognise if you have an obvious clinical symptom like this, which sticks out like this little frog here, but can be quite difficult to see if let me just see if I can get my pointer here. Yep. So, it may not be obvious even to clinicians who are trained in it to see really what the clinical symptomatology is like. Or if you like, a different way, mitochondrial syndromes can stick out like this tip of the iceberg, but there are many different symptoms that can lay hidden and be disguised. And so really, historically, we've only recognised just the tip of the iceberg, and we need to become better at diagnosing these disorders, because even though patients may not have severe disease, they can still pass on the disease, which can then become very severe and life-threatening in their children. Okay, so we developed a project really which recognised this problem with the broadening spectrum of mitochondrial disease, undiagnosed mitochondrial disease, and we really wanted to shift the diagnostic paradigm to improve and shorten and simplify this way that we could recognise mitochondrial disease in the clinic. The current diagnostic pathway is as such. It's reliant on clinical assessment, which can be extensive. Often this leads to the justification of a muscle biopsy, which is examined either histologically or biochemically, or both. We then do targeted DNA genetic testing, which might involve testing of the mitochondrial genome or the nuclear genome. And at the moment, the diagnosis lies between 25 to 50%. Slightly frustrating and not really accurate enough. What we proposed in this, in our new diagnostic paradigm, was to still keep the clinical assessment. I'm a clinician after all. Then to work on a series of biomarkers, which I'll talk to you about in a moment, and then to use these next generation sequencing techniques to identify the genetic diagnosis precisely so that we can personalise the medicine, the treatment for each of our patients. And we predicted that we would get up to about 70 to 80%. So I'm going to talk to you about the first diagnostic indicator that we studied. This was called FGF21, or fibroblast growth factor 21, and we found that it was a sensitive predictor for mitochondrial disease. We prospectively collected serum samples from 54 patients who had mitochondrial disease who came to our clinic, and we compared these to control samples and also neuromuscular disease samples. Patients have neuromuscular disease, sorry. Okay, what is FGF21? Well, it's a metabolic regulator. Essentially, it controls the way you release your energy supply. If you're starving, it will mobilise stores. If you're in a fed state, it will actually store away energy. And so you can see that patients with mitochondrial disease who have a loss of energy, they have a pseudo-starvation state. And so FGF21 is elevated in this situation. That was the hypothesis anyway. So here you can see the data. We performed serum levels of FGF21 in disease controls with neuromuscular disease and the patients with mitochondrial disease. And you can see here that there were significant elevations in FGF21 when compared to both controls and also patients with neuromuscular disease who may be masquerading as having mitochondrial disease. There was no difference between those patients that we diagnosed genetically or with muscle biopsy, and there was no significant difference between those patients who had proximal muscle weakness or who did not have muscle weakness. While we were doing this study, another biomarker was published called growth differentiation factor 15, and there was a proposal that this was a useful biomarker for mitochondrial disease. In this paper from the Japanese group, they measured FGF21 samples in 48 patients with mitochondrial disease. So what is GDF15, or growth differentiation factor 15? It's a member of the transforming growth factor beta superfamily. It's widely expressed in all human tissues, and it's secreted as a dimer and is present in body fluids such as blood and CSF. And essentially, it expresses, it's upregulated in a stress response, whether it be induced by hypoxia or anoxia or inflammatory conditions. So there are a number of drivers of GDF15, but one of them, you can see here, inflammatory changes, UV light, hypoxia, but once you get mitochondrial dysfunction, then you get elevations in GDF15. So we decided to look at that in the same population that we studied. Sorry, just to say that this is an indicator, therefore, of a stress response. And so we decided to do a comparison to see whether FGF21 or GDF15 was better in our patient population. So here you can see the comparative analysis. GDF15 here is coloured in green, and you have FGF21 in red. And essentially, the levels of GDF15 were elevated in all of these three study groups. And again, GDF15 levels were elevated in patients with mitochondrial disease and significantly elevated to indicate mitochondrial disease in these subjects compared to disease controls and also control levels. When we did the ROC curve analysis, comparing all of these techniques, we found that FGF21 had an area under the curve of 91, difficult to improve on, but GDF15 managed to be slightly better, significantly better, at 94%. The combination of putting them together, FGF21 and GDF15, did elevate the sensitivity and specificity a little bit better, but it's hard to tell whether this is significant, statistically significant with the numbers that we have raised from 94.1 to 94.4. But nevertheless, much better. And so we propose then that in this current diagnostic pathway that one should still start off with clinical assessment. If we use biomarkers to indicate who justifies whole genome sequencing, we can then move on to this whole genome sequencing process and then again possibly lead to 70 to 80% diagnosis. So we moved then on to next generation sequencing to see whether that was accurate in or to be able to use in diagnosis. And this is one of our first results. This was sequencing the entire mitochondrial genome in one run. So as you can see here, the mitochondrial genome is represented by this whole long sequence. You can get this in one piece. And if you haven't spotted it here, this is the mutation here that causes an ARP syndrome at 8993. But that was just looking at mitochondrial genome. So we wanted something a little bit better than that because we know that, that patients with mitochondrial disease may have genetic mutations in either the nuclear genome or the mitochondrial genome. Phenotypically, this was very difficult to determine at the bedside. So we progressed on to using next generation sequencing techniques, and this is the Illumina X10 machine, which is a series of 10 sequencing machines, which is able to cover the nuclear genome 30 times for every part of the nuclear genome, and we get very good coverage of mitochondrial DNA at the same time. This process generates 1.8 terabytes of data per human genome, and we can generate this within 3 days. So this is the machine being installed at the Garvan. These are our close collaborators who have the asset, the big sequencing machine, and you can see it here in its nude state. It's very rare to see it all uncovered here, so I just thought I'd show it for you. Okay, and what does the data look like? So again, it changes its appearance again. This is a patient who has POLG mutations. You can see here that we've sequenced the POLG gene. And this, in this format, you can see the mutations, the heterozygous mutations here and here. Again, this is a bioinformatic process, but it identifies the mutations at this point here and in chromosome 15 of the POLG, which is where the POLG gene resides. More importantly, we were able to analyse mitochondrial DNA by using different algorithms and software programs than you do for the nuclear gene. Remember, you won't get this on your normal analysis because you have to shift the filters to be applicable for mitochondrial DNA. But because the genetic material is amplified in a ratiometric way, we actually get 20,000 times coverage of the mitochondrial genome because there's a high copy number within the starting material. You can see here at this point that this is where 3243 resides, and in this sample we found that there was 15% of the 3243 mutation in this sample, which would not have been picked up by Sanger sequencing or other traditional techniques. And yet, therefore, you really do need to have high levels of coverage to be able to detect this sort of mutation, particularly in the start material that we have, which we used was being blood. So this is representative of the investigations that we perform at our clinic to try and diagnose mitochondrial disease before taking this approach. It included a complex algorithm of clinical testing, ophthalmological review, imaging, spectroscopy, ECGs, radionuclide scans, electrophysiology, genetic testing and muscle biopsy. And we now can simplify this down to a single blunt sample, which is then separated into biomarker studies and genetic testing. So now, just to summarise, this means that we can just to remind you that mitochondrial disease presents a diagnostic challenge, as it can present in many different ways and be under-recognised in the community. Next-generation sequencing can accurately identify causative mutations, and if we use whole genome sequencing, we're able to do this one-stop shop process of identifying both nuclear gene mutations and mitochondrial DNA mutations at the one time. And serum FGF21 and GDF15, we're also working on other biomarkers at the moment, combined with next-generation sequencing, may simplify the way that we diagnose mitochondrial disease, and I believe, in fact, in all other neurological disease, and negate the need for model investigations which may be invasive, such as the diagnostic muscle biopsy. So I'd just like to acknowledge my collaborators. These are the bioinformaticians that are working with the whole genome sequencing project. Ryan Davis is also a molecular scientist who is working on functional studies so that we can determine the pathogenicity of some of the functional variants. Our clinical team here, and of course in the bigger project we're working with the Australian Genomic Health Alliance to determine whether this can be moved into clinical service from an economical basis. Thanks very much.
Thanks, Carolyn. It just goes to show how bench to bedside is really moving forward. We've got time for a couple of questions, and if I could just start before Tony starts, you said you did the whole genome sequencing in 3 days, is that right?
I can do one genome in 3 days.
And the cost?
So good question. So research whole genome sequence is $1,600 at the Garvan. We don't charge that, but you do have to do the analysis yourself. I believe it's coming onto a commercial NATA-approved line. That's also available now, but the cost is more because you get the analysis.
$1,600 for a do-it-yourself.
That's right. I think that compares, if you want to do the whole exome study, that's really between $700 and $800 as a research price, and a mitochondrial whole exome is something around the same. So it's similar in price if you're looking at it on a research basis. You just have to spend a lot more time looking at the data.
Tony.
Yeah, thank you. You had heteroplasmic load on one of your slides, so you get all of your mitochondria from your mother, that's correct, isn't it? And therefore some of these mutations will only be in a subset in terms of that mitochondrial load. So what's the relative contribution in terms of somatic distribution of the mitochondria during embryogenesis and inherited versus somatic mutations of nuclear versus mitochondrial genes? And are you able to ascertain that?
Yeah, that's a great question. Trying to determine the difference between somatic mutations and those that are pathogenic is one thing. Obviously, so somatic mutations occur randomly. They're in a much lower levels of heteroplasmy, and they're scattered throughout the genome. And so, whereas heteroplasmic mutations can be slightly higher than the levels of heteroplasmy that you get, although it is sensitive, and we can detect in our hands levels down to 1% of heteroplasmy in patients with mitochondrial disease. So there has to be, this is why the clinical assessment is necessary, because you really do need to be able to interpret whether the mutation that you're identifying down to low levels of heteroplasmy are actually causative of the disease. So so in terms of volume, that's one way that you can determine between a somatic mutation and a pathogenic primary mitochondrial DNA related disorder. In addition to this, of course, the clinician's role is to then work with the bioinformaticians, because we then go ahead and do a process of tissue segregation. So in a somatic process, these are random again, but you might find we might repeat the mitochondrial DNA analysis in another tissue. So we start off with blood at the moment, but then we can use all sorts of tissue. Hair follicles is one, but but urine is also a good one to use at the bedside. So we traditionally collect paired samples in the clinic so that we can look at the heteroplasmic load in the urine as well. And you can do multiple tissues, skin biopsies, you can take buccal smears and so forth. And again, it's about trying to see whether it segregates with disease. For the pathogenic mutations, we don't do that, but for those that we don't know are pathogenic, that's the process that has to be followed.
Okay, well, I think we're out of time. Carolyn, thank you very much. Please join with me thanking you for a wonderful work. And now for one of our big events, I'm going to ask Andrew Allen to introduce the Fred Mendelsohn Lecture.
Thank you. This is a lovely opportunity to be able to honour two people at different ends of a medical research career spectrum. And I think it's a wonderful opportunity. I'd like to thank the other members of the organising committee for giving me the opportunity to make this introduction. So as a means of providing an opportunity to recognise and honour an outstanding graduate student in the neurosciences, the organising committee decided to inaugurate this student lecture and to give it during this symposium. The opportunity to deliver a lecture to this forum in association with the other high calibre speakers that we have here today is, I think, a rare honour and a rare chance for a postgraduate student. So I think, and we anticipate that this will become a sought-after award in the years to come. So from a list of submitted applications, 3 finalists were selected. They were given the opportunity to then present their lecture to the panel, and you'll get to hear from the winner in a few minutes, and we're going to honour all of the finalists after the lecture. So a substantial award, I think, requires a substantial name, and it's truly fitting, I think, that this award is going to be named after Fred Mendelsohn. And I'm going to try and generate a little bit of a picture to make the case for why this should be named after Fred. It's going to be anyway, but I'll do my best to make a case. So Emeritus Professor Fred Mendelsohn, Officer of the Order of Australia, General Division, Fellow of the Australian Academy of Sciences and past Director of the Florey Institute. These are but a few of the numerous awards and prizes that have been bestowed upon Fred over a lifetime of significant achievement. The Lectureship Award we're naming in honour of Fred is to acknowledge excellence in an undergraduate neuroscientist. And so, as I said, to try and make the case, I'm going to draw attention to just 4 areas of Fred's contributions that might inspire and cause people to strive for more in their research careers. Firstly, research. Obviously, we're here because it's research, and if Fred didn't have a stellar research career, we wouldn't be talking about him. Fred has written in excess of 300 publications. They've been cited well over 10,000 times. He's been invited to present this work to over 120 international lectures alone. His research has been instrumental in changing our view of one of the key humoral systems, the renin-angiotensin system, from an idea of it being a peripheral humoral system into a tissue-based system with a wide range of activities in a number of organs, including the one that's close to our heart, or in terms of this audience, in the front of our mind, the brain. We tend not to think of angiotensin as a brain regulator, but it is, and it has powerful effects within the brain. In addition, he's gone on to show that this system not just produces angiotensin 2, the renin-angiotensin system, but it produces multiple ligands with multiple effects. In the process, he's trained and mentored many investigators, some of whom, such as your current director, have developed distinguished scientific careers of their own. The discipline. Andy Lawrence, I'm not sure if he's here, once said to me, and I'm going to paraphrase him and I'll probably make a mess of what he actually said, but he said to me that in relation to the organisation of the Australian Neuroscience Society, that many people are very happy to participate in society life. They want it to happen, but very few actually contribute and give their time to the different aspects of a society. For a healthy discipline, we require this. Well, as a member of many journal editorial boards, as a member of the National Heart Foundation Cardiovascular Health Advisory Committee, National Health and Medical Research Council, including an important review of the sector through the Wills Report, and various other aspects, including being president of the Australian Neuroscience Society, I think we can very well conclude that Fred contributes and has contributed enormously to many aspects of his discipline. In terms of the Institute, my third point, you know Fred for his role as director of the Florey Institute, a role he accepted in 1996 when it was the Florey Institute of Experimental Physiology and Medicine. Fred was instrumental in focusing the issue exclusively onto the Institute exclusively onto neuroscience, and a direction that we can all be very thankful for today. Fred was pivotal in securing the agreement of the Florey Institute Board and the University of Melbourne to seek funding for a major expansion of the Institute into new buildings on the Parkville campus and the Austin Hospital. He worked with several others, many of whom are in the audience today, to bring many Parkville neuroscience entities into this building, the Stroke Research Institute, Mental Health Research Institute, and elements of the University of Melbourne Neuroscience community. I think this is a fantastic achievement. All of this occurred after Fred made major contributions in other elements of university life, He was a professor at the Department of Medicine at the Austin Repatriation Medical Centre, and that's where he established for the first time a research PET imaging facility in this country. Another significant achievement. Finally, I want to just draw attention away from a sort of traditional focused academic career. And the example that Fred demonstrated, that as academics we must contribute to broader society in its multiple facets. And whilst our research endeavours are important, it's also important and imperative that we raise our eyes above our little areas to look at the horizon. And a key example of this is Fred's involvement in the Medical Association for Prevention of War. He's played a pivotal role in this organisation since its inception, or soon thereafter, in the early 1980s. This is a society whose key charter is to work for the elimination of all weapons of mass destruction and the prevention of armed conflict. The Society's initiated a campaign to abolish nuclear weapons from the planet, and this has led to a campaign that's reached into nearly 70 countries. Its recent activities at the United Nations, involving the majority now of the world's governments, lend us some hope that substantial measures to reduce nuclear weapons will actually come into effect. This is key, this is important, and it's for us to remember that we need to look into the broader community and contribute where we can. So I hope I've made my case. We are honoured and thrilled that Fred has agreed to have this lecture named after him, and I think we should just thank Fred for his contributions. So with that as an introduction, it's now my pleasure to request that Matias Maturana comes to the podium, and he's going to be our inaugural Mendelsohn lecturer, and he's going to give us a lecture entitled Next Generation Bionic Eyes, Identifying Patient-Specific Spatio-Temporal Interactions in the Retina. Matias.
Thank you very much for the introduction there. My name is Matias Maturana. I'm a PhD student here at the University of Melbourne. I've been doing my PhD since 2013 with the Department of Electrical Engineering and the National Vision Research Institute, where I've been looking at improved stimulation strategies for the bionic eye, and I'm going to talk to you about some of the developments we've made in that space today. So I'd like to begin just by talking about how normal vision works. We're working in the visual system. So this is a cross-section of the eye. In the normal vision, light enters through the eye, through the cornea. It is focused through the lens onto the back of the eye on a very important piece of tissue called the retina. This retina is incredibly thin and delicate, but it does an incredible amount of processing of that visual information before it sends it through to the brain, and we see this as vision. We're going to focus onto the retina at the moment on a cross-section of this section here. And we see that the retina is broken up into three layers of cells. Light travels through the retina before it converges onto the photoreceptor cells. And these photoreceptor cells are responsible for converting light into a neural signal, which is passed through to the retinal interneurons and to the output cells, the retinal ganglion cells. And these cells are the ones that form the optic nerve and send information to the brain. So with diseases like retinitis pigmentosa or macular degeneration, these diseases affect the photoreceptors here. And through the progression of the disease, they die off and the patients lose the ability to see. However, the retinal interneurons and the retinal ganglion cells survive in large enough numbers that we can stimulate these cells artificially. And we can do that by implanting a bionic eye. The bionic eye has been under development for the last 20 or so years. There's many different groups around the world, including us here in Australia, who are working on different versions of the bionic eye. But they all kind of follow the same sort of architecture. They all have a camera that captures the visual scene. A body-worn processor will process the series of images and send a series of pulses into an implant that's implanted into the retina. And this is an electrode array which has a number of electrodes which can be activated to stimulate or excite retinal ganglion cells and artificially send information back to the brain. So here in Australia, I've been fortunate enough to be part of Bionic Vision Australia, the group that has been working on a bionic eye I've been part of it since 2011. What you see here on the right is an OCT scan showing the bionic eye, or the retinal implant, placed below the retina, which you see here on top. And looking into the eye on the left, you can see these nice round electrodes, and we can activate these electrodes to stimulate those cells, and activate these electrodes in a spatially relevant way to send spatially relevant information to the visual cortex. To give you an idea of what bionic vision actually looks like, I've made a simulation. And what you see in this simulation is these little spots of light. They're called phosphenes. The phosphenes are what patients have reported seeing when stimulated electrically with a bionic eye, and we can make many of these phosphenes appear to make a very simple phosphene image or phosphene shape. I want to point out that this is very much idealized, and it gives you an impression of where we want to get to in the future with the future generations of bionic eye. But there's a few reasons why I've made this simulation. And the first thing I want to point out is that I've taken a very detailed video and downsampled it to 300 spots of light, 300 phosphenes, and yet you can still convey a lot of the detail of the underlying image, and especially the moving parts of the image are much clearer than the static parts. The other thing I want to point out is that to convey this very detailed image, you need to be activating many, if not all, electrodes simultaneously. And this is something that the bionic eye does not do. The bionic eye only activates one or few electrodes at a time, mainly due to the unpredictable nature of how retinal ganglion cells will respond to multi-electrode stimulation. And this is one of the things that we're trying to address with our research. So I mentioned that the phosphenes in that video was very much idealized, and that's because every patient experiences phosphenes very differently. What you see here are some phosphene images drawn by some of the patients that were implanted here in Melbourne in 2012. Now, there was a series of tests run with those patients where a single electrode was activated and they were drawing out in space what they were seeing. And there was a finger tracker placed on their finger which could record their movements. So what you see on the left is a nice round phosphene similar to that video I showed you just before. This is when one electrode was stimulated. And then on the same patient, when a different electrode was stimulated, they saw this pattern here, which they've described as appearing like a duck. Now, the way that the current bionic eye works is in a very much open loop fashion. We have an image that we try to convey. Let's say we're just trying to activate a single electrode. And we convert that into a series of pulses, which will then excite a population of cells. And the patient will see this as some sort of image. And they've described things like donuts, ducks, hot dogs, bananas, quite a range of food groups, but not necessarily the actual information that we're trying to convey. So we're trying to close this loop. We would like to record the activity of those retinal ganglion cells and then use that activity to regulate the amount of stimulation that we apply in here. So for example, if we have a target spike rate that we're trying to achieve in a population of cells and we realize that we're not quite achieving that spike rate, through this algorithm we'll be able to change the amount of stimulation in order to try and achieve that target spike rate. And by this way, regulate the amount of stimulation automatically based on those patient responses in order to convey some more useful information to the patient, like helping someone find their keys or find their duck. So my talk today is very much what's being published in this publication here, where we've made three main contributions to bionic vision. The first one is that we've come up with a way of simultaneously activating many electrodes all at once. And while there are some groups looking at doing this, until now there's only been one publication that's shown how to predictably activate up to 3 electrodes at a time. We're looking at the activation of the entire electrode array in a predictable way. We've also come up with a mathematical model that relates the responses that we observe in the retinal ganglion cells to the stimulation that we've applied. And mathematical models do already exist as well, but they tend to be really complex models like the Hodgkin and Huxley model, which are not suitable for closed-loop control. Our model is suitable for closed-loop control. And the last point is that we've been able to recover the spatial temporal electrical receptive fields. And I'll describe these more in a moment. But it's a description of how electrical interactions occur in the retina to give rise to both excitation and suppression in the retinal ganglion cells. So a typical experiment in my lab would look like this, where we have these large electrodes in the background that you can see here, where I have explanted a piece of retinal tissue from a Long Evans rat and placed it on top of this array. You can see some lycra threads that are keeping the retina in place. And we have a patch clamp pipette here, a very sharp glass tube, which allows us to record from the inside of a single cell. And we can fill that pipette with a dye that allows us to recover that cell in 3D afterwards, and that's exactly what you see here. This is the cell that we've recorded from this very experiment. And what we're trying to do is relate the responses that we observe in those cells to the stimulation that we're applying. And we're always applying a biphasic pulse, so a positive pulse followed by a negative pulse or vice versa. And we do this to protect those electrodes from corrosion, but also to protect the retina from damage. So we're applying stimulation at constant frequency. We're exploring 10, 20, and 30 hertz. And the amplitude of stimulation is completely random. It's taken from a Gaussian distribution. You see here an amplitude of level A or negative A has the same probability of occurrence, which is quite low compared to amplitude closer to 0. We use this stimulation because it has some very well understood statistical properties, and really the main one that we're interested in is that the stimulus amplitudes are completely independent in space and time. So what that means is that for any given electrode, the amplitude that we apply on that electrode is completely independent from every other electrode and completely independent in time from every other electrode as well. However, if we look at the select few stimuli that are generating a response in the neuron, we should find that these select few stimuli are no longer independent in time or space. For example, for a given neuron, we might only expect that electrodes close by to that neuron will affect its response. Distal electrodes will not affect its response. And maybe we might find those sort of correlations in time where electrical stimulation occurring over time might influence the response as well. So we're looking at a time window of 300 milliseconds, so stimulation applied over that window. And to explain this in a bit more detail, here is a cell that we've recorded from. We presented more than 50,000 stimulus patterns to this cell. This gives you an impression of the stimulation that we're applying, completely random, 10 hertz stimulation, and we've recorded more than 10,000 spikes for this cell. So just intuitively, you might expect that for this cell, electrodes 6, 10, and 11 will influence its response. So we can look at the amplitudes that we apply on electrodes 1 and 2 and plot it into this graph here, where you see a negative amplitude on electrode 1 and 2 corresponds to a point roughly here. A small positive amplitude on electrode 1 and a large positive amplitude on electrode 2 corresponds to a point here, and on so. The stimulation is limited to plus or minus 300 microamps, which is the limits of our stimulator. And we see that this distribution of input stimuli looks very Gaussian, as we expect. We have high density close to 0, lower densities moving off to the higher amplitudes. Now, we can colour in a subset of these stimuli in red, just the ones that are generating a spike in the neuron. And if we do that, we see that the two distributions, the ones that are generating a spike and the underlying input stimuli, are pretty much the same. They're both looking very Gaussian. And what this tells us is that electrodes 1 and 2 are not influencing the response of the neuron. But what if we do this for electrodes 6 and 10? Here we see something really interesting happening. The distribution is clearly not Gaussian anymore. We see that electrodes 6 and 10 are having an excitatory response in the neuron, and we can tell that by realizing that as we increase the amplitude along electrode 6, we have a higher probability of generating a spike. Similarly, for decreasing in the high negative amplitudes along electrode 6, and similarly for electrodes 10. So this is a really good start. It tells us that electrodes 6 and 10 are having an excitatory effect on this neuron, but it's not the full story. What we would like to know is how all of these electrodes are influencing the response of the neuron. And we can actually find the combination of electrode amplitudes that maximizes the variance of these red dots to find out how they're all influencing the response. And we do that by comparing the statistics of the input stimuli, which was Gaussian, and the statistics of just the ones that are generating a spike. Now, this stimulus pattern here that you see here is the one that is most influencing the neuron's excitatory response. And we see, as I had postulated at the very beginning, it's these three electrodes that are influencing the neuron's response. Sorry, I'm afraid one equation's not coming up here, but I'll try and describe what's happening here. This axis here, we're now plotting the input stimuli along this axis here, which represents the dot product of all of the stimuli onto this component here. So this is a stimulus. Essentially, it describes a stimulus. And by doing the dot product, what we find is that if the stimulus that we're applying is very similar to this one here, we'll get a high positive value for the dot product. It's like a correlation coefficient. It basically tells us how similar a stimulus is to this one here. If we have a stimulus that is not very similar to this one here, we'll have a very low value for this dot product. And if we have a stimulus that is very similar to this one here, but of opposite polarity, we'll have a very negative number. So that's this axis shown here. And if we see the red dots, the stimuli that are just generating a spike in this neuron, we can see that it's clearly bimodal compared to the input stimuli, which is Gaussian. And by comparing the histograms of the input stimuli, which we see here, the white one looks very Gaussian, as we expect. The red one looks very bimodal. We can compare the ratio of these two histograms to find a spike probability. And that's shown here. We see that the spike probability tells us if we're stimulating along a component that is, or along a stimulus that is very similar to this one, we have a high probability of generating a spike in the neuron. And if we're stimulating with something that's not very similar to this one here, we have a low probability of generating a spike. I'm sorry for the axes, I'm not sure why they're not appearing here, but I'll try to describe what's happening here in as much detail as possible. But basically, this gives us a relationship now that says that the response of a neuron, given an arbitrary stimulus S, is the dot product of that stimulus with this stimulus here. And basically, if the stimulus is very similar to this one here, we expect a high probability of response. If it's very dissimilar, we expect a very low probability of response. Now, just to point out, I mentioned before that we're looking at a time window of 300 milliseconds, but what I've shown you here is only the spatial component of the electrical receptive field. It doesn't show any temporal correlations here. And that's because this neuron was stimulated at 10 hertz. We actually found that for most neurons that were stimulated at 10 hertz, there was no temporal correlation. There was no integration of stimulation over time. Now, just to revisit this example that I've just described, in this case here, we're trying to find a combination of stimulus amplitudes that maximize the variance of the red dots, the ones that are producing a spike. We can actually do the opposite and find the combination of electrode amplitudes that minimizes the variance of the red dots. And that gives us suppression. And we see that if we do the same analysis, this is now plotting it against the suppressive component, we have clear suppression occurring in this neuron. If we're stimulating at high amplitudes along this axis here, we have a clearly reduced spike probability or response probability in this neuron. Now, to show this in a bit more clarity, here's an example of a cell that was stimulated at 30 hertz. Similar to the previous example that I showed before, the excitatory response is shown here. This is time preceding response going all the way to 300 milliseconds, but I'm only showing up to 66 milliseconds in this case. We see that there's no temporal correlation for the excitatory component in this case here. This is the stimulus that the neuron is most sensitive to for excitation, and this is the stimulus pattern that this neuron is most sensitive to for suppressive stimulation. And we see something very interesting here. We see that they're very, very similar, except this one's shifted back one stimulus frame. And what that tells us is that if we excite this neuron, there is a very good probability that we're going to suppress its response for 33 milliseconds thereafter. And I want to point out that this is not just a refractory period effect. This is happening on a much longer time scale than refractory periods. So what this is actually telling us is that we're modulating the retinal interneurons in a way that's either causing excitation or suppression in the neuron's response. And we found that for cells stimulated at 20 and 30 hertz, the suppressive components could last anywhere up to 100 milliseconds. So in total, we recorded from 80 retinal ganglion cells. For each cell that we've recorded from, that equated to about 3 or 4 hours of stimulation. And we presented each cell with anywhere between 50,000 and 150,000 stimulus patterns. And for each cell, we used 80% of the data to find the model parameters, the nonlinear function and the excitatory and suppressive components, and then the remaining 20% to test how well our model performed. And that's what you see here. On the x-axis is the predicted number of responses, and on the y-axis is the actual number of responses. And you can see that for all of the cells, there's very good correlation between our prediction and our actual number of responses. So in conclusion, I've shown you today a mathematical model which we can use to predict how retinal ganglion cells will respond to arbitrary patterns of stimulation. And importantly, this allows us to stimulate with the entire electrode array in ways that we can actually now exploit all of the electrodes that we have available to us to make some more detailed phosphene images for the patients and hopefully more functional vision for them. I've also shown you how spatial temporal interactions occur in the retina and the way that we can actually find these spatial temporal electrical receptive fields. And quite importantly, by understanding how electrical stimulation is leading to both excitation and suppression in the retinal ganglion cells, we can actually control for these effects in a closed-loop control and actually exploit these effects in more useful ways, with the hope that in the future we'll be able to generate some more useful phosphene images and be able to help people recognize someone's face, for example, something that many of us take for granted. So before I finish, I'd like to thank my supervisors at the National Vision Research Institute and Electrical Engineering. A special thanks to Nick Apollo for fabricating the devices that we work with, and a big thanks to the organisers for today for giving me this opportunity to talk. Thank you very much.
Thank you, Matias. We've got some time for questions if people have them in relation to this presentation. Jeff?
So could I stress that what I understand is that this is going to modify every one of those pixels so that you get you might downplay some pixels and you might upplay others.
Yes,
so that
yes.
You get a clearer image.
That's right.
And so at what stage of development is it now? Have you got images that you've got, say, without the feedback, and then images with the feedback to show there's a distinct difference?
So we haven't gotten up to the feedback. I mean, up until now, my PhD has been very much focused on recovering these electrical receptive fields. And in my postdoctoral work, we're now exploring much smaller electrodes, so hundreds of electrodes, and being able to do this on population of cells as well. What we've done as well is a combination of this wet lab experiments and also simulated works where we've been able to simulate how we can control for these responses, and that's where we're really looking at applying those algorithms in my postdoctoral work at the moment. So yes, we haven't actually compared the closed loop and open loop responses yet, apart from simulation. Yeah.
An obvious way to increase resolution would be a higher density electrode, right? But if you do that, would lateral spread of current diminish the effectiveness of the obviously I've got much lateral spread at present to show that surrounding cells.
Yes, so I would expect that the higher density will actually improve our responses because we'll be able to focus electrical currents and actually probably even steer. I mean, the hope is that we're able to steer currents between electrodes in order to target specific areas of cell or specific areas of the tissue. This is something that we haven't been able to demonstrate with these very large electrodes. We've used these large electrodes because it has a direct clinical application in future studies where we implant more patients. But we are looking at moving to smaller electrodes, and we hope that with that, well, we know that retinal ganglion cells have a light receptive field, and they possibly have something very similar in terms of the electrical receptive fields as well. So there might be some sort of relationship that we can exploit there as well with the smaller electrodes. Yep.
Go there and then there. So,
yep.
I guess this sort of stuff is quite computationally demanding when we're real time and firing where it's computer on your back. How quickly is computational stuff keeping up with this kind of technology, and are you developing something that's not really going to be feasible in the future to go to higher density arrays?
That's a very good question, and actually computational time is a very critical factor for this. From our simulations at the moment, we've found that computation for, well, we need to predict what the neurons are going to do before we apply the stimulation, and we have to predict that in a very small amount of time. So computation for that has taken anywhere up to 50 milliseconds, which means that we'll be limited in terms of our stimulation frequency. But this is very naively, doing computational very naively. So if we were to do parallel computing or had software or hardware that enabled us to do that, we might be able to improve on that and get to the higher stimulation rates, which would be really useful for us. At the moment, yes, just naively, we're limited to roughly 50 milliseconds of computation time. Yeah.
Can I quickly follow that up?
Yep.
Also, taking into account you're trying to replicate what neurons are doing with the electrical stimulation, so can you also take into account like refractory periods and do what neurons would do in that situation?
So, well, because we're stimulating at, I mean, the highest stimulation frequency that we've looked at is 30 hertz stimulation. we were kind of well beyond the refractory period by then, because refractory period for retinal ganglion cells, I mean, retinal ganglion cells can fire up to 300 hertz roughly, so quite fast cells. So we're well beyond the refractory period there, so we're not really taking into account any refractory period effects there. We're only taking into account the modulation of those retinal interneurons in a way that's either causing inhibitory effects or suppressive or excitatory effects on the output cells. So, yeah.
Last question.
So I guess I missed what was limiting currently, other than just where you are in the project, the number and intensity, size of the electrodes, treatment down. This is a related question. So it's possible that you'll never get to the stage where you'd even want to mimic the patterns of action that is coming out of retinal ganglion cells. But you might, in a human being, retrain them on a visual world that gets transformed with that, right? And so you guys started experiments like that. So you take what you can do to the retina, and you pick a representation that you can then transform, and a person can learn to interpret that. People are quite flexible about these things. You can flip the visual world into
Absolutely. Yes.
And things like that.
Yep. Well, we haven't explored that, and that's kind of probably beyond our scope. It is a very interesting question, and it is something that a lot of researchers are looking into. What we hope to do is actually more recreate those actual action potentials from visual stimulation in something similar, at least. Not exactly, but something similar at least. I guess that's where our focus is more is on at the moment. So if we can, for example, stimulate the retina with light and try to create or record those activity patterns and try to recreate them with our predictive models as well, that's where we would be looking at the moment. Yeah.
Well, thank you very much, Matias. So if I can ask Fred to come forward, Matias can stay here. This is the award ceremony part of it, and I'm hoping someone's going to come forward with some sort of award at some point that is over there. And so that's probably me coming over here. You need something. No. Right. Okay. So, here we are. So, first prize, clearly, goes to Matias for his wonderful presentation that you've heard, and Matias and Fred. Congratulations. These are quite remarkable. So, this is the second prize, and this goes to Dean Wright. And Dean Wright, his presentation topic was modulating glutamate dysfunction in Huntington's disease using N-acetylcysteine. Third prize, now is Daniel here? Oh, Daniel is here. So third prize goes to Daniel Bennett for the topic, A Need to Know, Neural Substrates of Information Seeking in Decision Making Under Uncertainty.